This week we were asked to do a presentation on ATP production. Glucose is the main energy source for cellular metabolism. Glucose is broken down in 3 stages, the first being glycolysis, where glucose is converted into pyruvate. The pyruvate is then converted to acetyl-CoA. Secondly is the Krebs cycle, where acetyl-CoA enables production of NADH. Third is the electron transport chain, where NADH is used by the respiratory chain complexes to generate a proton gradient across the inner mitochondrial membrane, which is necessary for production of a large volume of ATP. Taking a closer look into glycolysis, we can see that this process occurs in 2 steps. The purpose of glycolysis is to break down the 6-carbon compound glucose into 2 3-carbon molecules called pyruvates. Phase 1 can be referred to as the investment phase, in which 2 ATP are required for this step. By the end of phase 2, which we refer to as the payoff phase, a total of 4 ATP are made, resulting in a net total of 2 ATP. Phase 1 is an irreversible step in which 2 phosphates are added to glucose. The phosphorylation traps the glucose molecule within the cell. Next, the phosphoglucose isomerase isomerizes glucose 6-phosphate into fructose 6-phosphate. Then the second phosphate is added by phosphofructokinase, which uses the second ATP and phosphorylates the fructose 6-phosphate into fructose 1,6-biphosphate. This is considered the rate-limiting step. In the payoff phase, 5 additional steps are completed, resulting in the formation of 4 ATP, 2 NADH, and 2 pyruvate molecules, ultimately resulting in 2 net ATP. These products then enter the Krebs cycle, which consists of 8 steps. Step 1, acetyl-CoA joins with oxaloacetate to form citrate. Step 2, citrate is converted to isocitrate. Step 3, isocitrate is oxidized to alpha-ketoglutarate, which results in the release of carbon dioxide along with 1 molecule of NADH. Step 4, alpha-ketoglutarate is oxidized to form a 4-carbon molecule, which picks up coenzyme A and forms succinyl-CoA and an NADH molecule. Step 5, succinyl-CoA is converted to succinate and 1 GTP molecule is produced. In step 6, succinate is converted into fumarate and a molecule of FADH2 is also produced. Step 7, fumarate is converted to malate. Step 8, malate is converted into oxaloacetate and NADH is also produced. This process occurs twice, leaving us with a total of 2 ATP, 6 molecules of NADH, and 2 molecules of FADH2. Stage 3 is the electron transport chain. This occurs in the cytoplasm in prokaryotic cells and in the inner mitochondrial membrane in eukaryotes. During this stage, an electrochemical gradient is formed via proton pumps. ATP synthase then uses subsequent diffusion of protons to synthesize ATP. Oxygen ultimately accepts electrons and protons to form water. By the end of this cycle, we are left with approximately 32 to 34 ATP, H2O, and NAD+. This slide shows the conversion from NADH and FADH2 to ATP, which theoretically yields approximately 30 ATP.